great pleasure that I, Shloka Roy, welcome you all to the International Seminar on Emerging Trends in Cancer Biology, hosted by the Center for Drug Discovery and Development, CD3, at Amity University Biotechnology. Today, we commemorate World Cancer Day by delving into the latest advancements and discoveries in the field of cancer biology. Cancer remains one of the most pressing health challenges of our time, affecting millions of lives globally. However, through collaborative efforts in research, innovation, and education, we strive to unravel its mysteries and develop effective strategies for prevention, diagnosis, and treatment. World Cancer Day, observed on February 4th every year, serves as a global platform to raise awareness about cancer, encourage preventive measures, promote early detection, and advocate for improved treatment care. It unites individuals, communities, governments, and organizations worldwide in the fight against cancer. World Cancer Day also provides an opportunity to celebrate the progress made in cancer research and honor the courage and resilience of cancer patients, survivors, and their family. Today's seminar serves as a platform to explore the cutting edge research, breakthrough technologies, and the emerging trends that are reshaping our understanding of cancer biology. We are honored to host renowned experts and researchers who will share their insights, expertise, and experiences with us. Thank you once again for your presence and participation. Without further ado, let us commence the International Seminar on Emerging Trends in Cancer Biology. To start with more than 30 years in the field of biotechnology and plant sciences, Dr. Penna Suprasanna is the Dean Research of Amity University, Maharashtra, and the Director of Amity Institute of Biotechnology. With a PhD in genetics from Osmania University and a publication count exceeding hundreds, Dr. Suprasanna's impact extends beyond groundbreaking research. He is the former head of Nuclear Agriculture and Biotechnology Division at BARC, Mumbai, and the former professor in Homi Bhabha National Institute of Mumbai. He is on the prestigious editorial boards of international journals and is a valuable member in many communities for development of science in agriculture. Through these collaborations, he has played a pivotal role in developing technologies that enhance plant life, growth, and resistance. He's a passionate educator. We are fortunate to have Dr. Penna Suprasanna with us today. So let us invite him for the welcome address.
Thank you, Sue. A very good afternoon to everyone and a warm welcome. Honorable Vice Chancellor, sir, and the organizers of this international seminar, and the guests for today, Dr. Shankari and Dr. Rizaz. I'm very happy to see that AAB is organizing such an event on the eve of World Cancer Day. 4th February was the date, so we are hosting the World Cancer Day to see the close the care gap theme that internationally has been put forward for the world cancer awareness. And as Shloka said, we are going to witness the current cutting edge technologies that have happened in the field of cancer care, cancer therapy, cancer management, and we have two experts who are going to deliver the talks. I'm very happy to see that AAB is organizing, and I welcome you all. Thank you very much.
Thank you so much, sir. Coming next, our Honorable Vice Chancellor, sir. His journey spans over 34 years, which reflects an unwavering commitment to academic excellence, leadership, and fostering a global outlook in educational endeavors. He holds a PhD from the prestigious All India Institute of Medical Sciences, AIIMS, and has pursued his postdoctoral fellowship in renowned institutions like University of California, Davis, the National Cancer Center of Singapore. His expertise lies in microbiology, infectious diseases, genomics, and he contributes significantly to these fields through his 180 peer-reviewed publications. Dr. Santosh Kumar's global outlook is evident in his collaborations with organizations such as the National Institute of Health, NIH, and his experience working in the United States for over 16 years. He actively promotes international collaborations and student exchange programs, fostering a global perspective among students and faculties. He has held various positions, such as Dean, Academic Affairs, Accreditation, Co-Director at Inland Stem Cell Consortium, Co-Director, Center for Genomics, Director at STEM Program, and the Professor in Microbiology and Infectious Diseases. It is my privilege to invite to the stage our Honorable Vice Chancellor, Sir, Professor Dr. A.W. Santosh Kumar, for his opening remarks.
Well, thank you very much. Elite gathering, respected Dean of Research and Director in Charge, Amity Institute of Biotechnology, the Head in Charge of Center for Drug Discovery and Development, the faculty members of the center, faculty members of Amity Institute of Biotechnology, my dear student friends, and the two elite speakers that we currently have, Dr. Sankri Nagarajan from University of Manchester, UK, and Elijah Ahmed from University of Michigan Medical School, USA. It's indeed a very great pleasure to be with you, being a medical microbiologist and a biotechnologist, heading this institution and being a part of this high-powered lecture in commemoration of the World Cancer Day. As you all know that the WHO has given a red flag stating that there will be 35 million cases that will be increased within 2050 with a 77% hike in the incidence of cancer. And as we all know that any one given point of time, our relatives or friends are just going through a very rigorous system of cancer affection or a cancer therapy or a cancer treatment or a cancer diagnosis. And we have two elite speakers who have done research and currently on the verge of doing the research in many different fields, and very young researchers, one from the UK, another from the USA, who now take part in delivering their own experiences in their research to our students of Amity University, Mumbai. I really thank the organizers for selecting these two speakers and the Center for Drug Discovery and Development for having remembered the World Cancer Day, and also to just commemorate. Since 2018, we have not gotten any Nobel Prize for cancer research or cancer therapy. And I hope that these kind of riches will kindle the cancer treatment to bridge the gap that is being given as the motto for cancer, World Cancer Day 2024. Thank you very much for having me here, and I look forward to the deliberations further on. Thank you very much.
Thank you so much, sir. With the inspiring words from our Dean and Vice Chancellor, sir, let us now embark on this enlightening journey of discovery and collaboration. Our first speaker, Dr. Ejaz Ahmed, is a distinguished scholar and researcher whose expertise spans from protein biochemistry, cell division, cancer biology, drug discovery, and high throughput screening. Dr. Ahmed's academic journey is complemented by extensive postdoctoral training and research experience at esteemed institutions such as the University of Toledo, Sao Paulo State University, Central European Institute of Technology, and the University of Michigan. Throughout his illustrious career, Dr. Ahmed has made substantial contributions in the field of biomedical sciences, as evidenced by his numerous publications, patents, and awards. He has been recognized for his exceptional research contribution with honors. Dr. Ejaz Ahmed has been recognized for his exceptional contribution to scientific inquiry and scholarship. For example, his pioneering research in protein-protein or drug interactions, groundbreaking discovery in cell division and gene expression, significant contributions to cancer biology and drug discovery, and there are so many more. Additionally, Dr. Ahmed serves on the editorial board and as a reviewer for several esteemed journals, demonstrating his commitment to advancing scientific knowledge and scholarship. With this diverse expertise and unwavering dedication to research and mentorship, Dr. Ejaz Ahmed continues to be a driving force in the field of medical science, inspiring the next generation of scientists and making significant strides in understanding complex biological processes. Now, please join me in welcoming Dr. Ejaz Ahmed.
Hello, can you hear me?
Yes, sir.
Okay. Hello, everyone. So thank you so much for such a nice and wonderful introduction. Really, I'm amazed. I know all the things are from my CV, but listening these things is really very means very joyful for me. Yeah, like I can say. So today I think I have to start now my talk. Is it? Should I start now?
Yes, sir.
Okay. Okay, so I am sharing my screen. Okay. So, as in introduction, has been already told all about me and my work. So today I am just going to talk one of my domain of research. So where we are working on the drug discovery against the cancer. And cancer, it is mainly leukemia. So, and we are targeting BCL2 family proteins and by small molecule inhibitors with not just one compound, but also other therapeutics in this field. So let us start further. So I was just in brief that what is cancer. So it is an uncontrolled cell division and growth. So what happens that in the normal cell, if there is some mutation, so which increase with time, so the type of mutation and number of mutation are increasing. So they increase too much and they become a tumor. So in this, in the cancer, what happens is actually this is a disease of altered gene expression. So this gene expression is because of these mutations. So and these genes, some of the gene because of this mutation can not express, can be lower expressed, or can be higher expressed. And because of this this altered expression, the quality and quantity both can be affected by the gene and gene product. And these alteration affects directly some protein interaction, which are key regulator of some pathway. So that pathway can be anything, metabolic pathway or whatever. There are multiple cell biological pathway. So there can be no interaction because of this mutation, and there can be more interaction or new interaction, which is not exist in normal cells. So this is a little bit brief cancer introduction. So I just want to go some current scenario on the global how the cancer affects in the at the global level. So yeah, here I am showing that incidence rate of different cancers. So I got these source from Globocan. So you can see here that leukemia is 32% of all the cancers of the incidence. And at the, if we want to see the continent level, that in Asia, it is highest. But if we are going to see the mortality rate, so we will again see that it is the leukemia, and so almost highest mortality rate. And in at the level of global, you can see that in Asia it is highest. So we can say it is very clear that the how some countries like Asian countries they are not just only incidentally, they are more incidents, as well as there is high mortality rate also. So, but there is, and again, one more thing, you can see at the global level in the map. So if you can see that at 100,000 person, how many incidents are there? So here, if you see in the in US, Europe, Oceania, these places, the incidence is too high. While in Asian, like India and some other Asian countries, the incidence rate is low. But if we see the mortality, so in US and Europe and other, and in these continents, these countries are, you can say, in the continent at all, in the North America and Oceania, the mortality is lower in comparison to Asian countries, where the incidence of leukemia was lower. So it is really very alarming situation that, and we should know that why it is happening, that, and it is very clear that what are the reason. No need to discuss here. So I will talk all about leukemia, how it is we are doing drug discovery and how we are trying to inhibit this disease. And so our lab, our group is working on anti-cancer drug discovery and mainly targeting protein-protein interaction. So, and in leukemia, there are, and we are targeting different ways, like at epigenetic level, where AF9 or ENL in interaction inhibited with DOT1L. So if we inhibit, so there will be effect in the methylation of the histone H3K79, and the gene for progression will be affected. And the other one, we are also validating the ENL East domain, how the gene rearrangement is affected. So we are also inhibiting, means discovering the inhibitor against MLL ENL for the East domain of the ENL. And the most means I am mainly involved in the third one, like targeting BCL2 family protein. So there are multiple type of BCL2 family proteins. Two examples are MCL1 and BFL1. So this is our work, and we are so we are getting some. We got already 2 compounds, but it's still under process, so it is not yet patented and something. But we are working on this one. So today I will discuss one more thing. So because I already told that we are discovering agents against these anti-apoptotic protein or BCL2 family protein. We are also doing the clinical trial assessment of already available BCL2 inhibitors with some known chemotherapeutic agents in leukemia patient. So as per introduction, I just want to say that what cells will die, what will not die, why they are dying, what not dying. So there are apoptotic proteins. Actually, apoptosis is programmed cell death. So which is required in the cell. So if there is wear and tear, and if the body needs to die, and the body needs to die the cells, then apoptosis happens. So for apoptosis, there are anti-apoptotic protein, pro-apoptotic protein. The balance, and there is a balance between these proteins. So what happens if pro-apoptotic protein are higher in expression, in function, there will be cell death. But if in opposite, if the pro-apoptotic protein are low or anti are high, it is not just expression, function, everything. If anti-apoptotic protein is high, the cell will survive, they will not die. It happens in case of cancers. So this our target is this, to inhibit this anti-apoptotic proteins. And this is because the cell's survival depends on this protein, like anti-apoptotic protein. So that's why we say the survival dependency of cancer cells. So this is our target. What we do, In drug, so how we use these protein interaction of BCL2 family proteins between anti-apoptotic and pro-apoptotic protein. So what we do, and that here you can see the characteristic feature of BCL2 family protein. So they all have BH domain. So from BH1 to BH4. Some pro-apoptotic protein also have, which are effector back and backs. But there are one other class of pro-apoptotic protein which are BH3 only protein. They have only BH3 domain. So if the binding of this BCL2 anti-apoptotic protein with pro-apoptotic protein, if they can be inhibited, then those drugs will be known as BH3 mimetic. Because so we can make that drug by characterizing the features of these pro-apoptotic protein domain of BH3. So these are examples of BH3 peptides. So you can see in the sequence alignment that this aspartic acid everywhere. So if we make some drug which has also aspartic acid, or so like aspartic acid, other residues, so that can inhibit these interactions. So one example is MCL1. MCL1 is one anti-apoptotic protein, and this is NOXA, one pro-apoptotic protein. So here, if at this place we can put some molecule, so they obviously NOXA or this pro-apoptotic protein will be out, and this will be used in apoptosis, and the cell will die. So this is the strategy how we are developing and BCL2 inhibitors. So BCL2 or BCL2 family, actually I will say this thing multiple times. So all these cells and these proteins, they are whether it is anti-apoptotic protein or pro-apoptotic protein, they are known as BCL2 family protein because of their domain. And BCL2 itself is a protein. So it's a little bit some confusing, but just want to let you know here that MCL1, BFL1, BCL2, so these all are anti-apoptotic protein. And one example I am giving of the BCL2, this BCL2 protein, it is bound with PUMA. So PUMA is a pro-apoptotic protein. It is a sensitizer like that. So what they did means, so this is the molecule which is known as venetoclax or ABT199. So this is the only compound or drug which is available right now on these days, and what it is the only FDA approved drug. There is no other drug for other BCL2 family protein, anti-apoptotic protein like MCL1, BFL1, BCL. There is no drug. Only BCL2 has one FDA approved drug. So how our strategy, that how we are developing our some inhibitor. So first we have to know that how these proteins like pro-apoptotic and anti-apoptotic, these proteins, how they interact. So for that interaction, to know those interaction, we are doing some like biophysical characterization of those interactions, so that we can develop an assay, and that assay can be used as a screening for the compounds. So here very first we used fluorescence polarization assay. So where we are taking one part of one part of the complex of the pro-apoptotic and anti-apoptotic. So see here, this peptide, we can say it is pro-apoptotic protein. So we labeled with some fluorescent probe. So actually in fluorescence polarization, what happens, the light which the incidence light or excitation light, they are unpolarized, which are which are through polarizer, we can change it into polarized light. So those lights, when interact with this small molecule or this peptide which with fluorescent tag. So it is very small. So what happens? There is a tumbling in this protein peptide and fluorescent molecule. The very high tumbling. Because of the high tumbling, the emission and the emission of the parallel and perpendicular, it is nullified. So there will be no signal. But if there is protein, so protein will bind with this peptide and there will be very big complex. So because of the bulkier size and bulkier size, the tumbling will be low and the only one kind of polarized light will be detected by the signal. So you can see here that it is only the perpendicular light is detected. So there will be signal. But if there is some competitive inhibitor which can bind here, then it will detach and again there will be no signal. So we are using this method for assay development. And so I can show you some data that these are anti-apoptotic protein BFL1, MCL1, BCL2, and BID is pro-apoptotic protein which can bind with all anti-apoptotic proteins. So FAM is fluorescent label molecule. So BFL1, MCL1, BCL2, you can see that a very nice binding curve with these pro-apoptotic protein. And if you add the inhibitor, you will not see this binding. So the other way, we are using some other methods after that, because one method is never enough for any experiment or something. So we are doing some other thing. So like surface plasmon resonance or BLI. So I will discuss here on BLI because I generated some data with this, which I'm going to show. So here one protein molecule, because I'm talking about the interaction, protein-protein interaction, and here apoptotic protein, pro-apoptotic and anti-apoptotic protein. So one partner will be bound with immobilized with this needle, and when the incidence light will be passed. So because if the difference in the thickness of this the tip of the needle where one protein is already bound. So if the other molecule will bound, the thickness will be high and then the wavelength will be changed. So this is the principle of BLI or surface plasmon resonance here. So I generated one data. So here you see BFL1 or A1, both are same name. So someone is saying A1, some say BFL1. So this was immobilized and FS2 is a pro-apoptotic protein. So you can see how clear binding you can see here. So if you have to develop some drug, just add that compound here. If there is no binding, means it is inhibiting the protein-protein interaction. Then and from this method, you can also know that what is K on and K off, which is very important for the drug discovery, because you must have to know that what is the kinetics of the binding. So then we are also using isothermal titration calorimetry to know that by thermodynamics of these interactions. So here this is the machine. So I can show you the internal structure of this machine. So what happens? So there are two kinds of chamber, one difference cell, one the sample cell. Here we add our one protein and then there is a titrator. So this is ligand. So it can be compound or other protein. So when there is some interaction between these two entities, so there will be change in the temperature and the instrument will be given another feedback power. So that is so you can see that thing and the signal of enthalpy change. So the upper panel is the raw data of the so it is showing the enthalpy and when it has been the data or area under curve has been plotted or it is analyzed. So you can see a curve. So the slope of this curve, this sigmoidal curve, the slope is giving us the binding constant, and from this slope and the 50% of this will give you the molar ratio, and this will be stoichiometry of the binding. So from this you can know that whether this binding is how many molecules are bound there, and you can also see the enthalpy. So it is uh, from these you can know that what is the binding constant, because Kb is directly inversely proportional to to Kd, and from this because you have the Kb, you are getting the free energy change. From free energy change you already have free energy and then enthalpy change, so you can get the entropy, and from this entropy you can get a specific heat. And the beauty of this instrument is that from the symbol of the enthalpy change and entropy change, you can tell that what kind of interaction are there, whether it is hydrophobic or ionic or hydrogen bonding. So these are the way how we know the protein-protein interaction. And here I can show you that all those interaction. So I can put that, I'm putting here that how pro-apoptotic and anti-apoptotic proteins bind each other. What happens in general, that if there is DNA damage and or chemotherapy is given, so the activator, which are pro-apoptotic, BID and BIM, they activate backs and back, and they dimerize, and they form ion channel, the channel in the membrane, mitochondrial outer membrane, and then cytochrome C release, and there is apoptosis, and this way the cell will die. But what happens that because of the DNA damage or chemotherapy, there is increment, very high expression of the anti-apoptotic protein. So BCL2, BCL, MCL1, BFL1, all those. So you can see that the BC here I have shown that how they inhibit or they bound with these pro-apoptotic protein. So all these protein can bind with each pro-apoptotic proteins. So if it will happen, there will be no apoptosis. The cell will survive. And the other pro-apoptotic, which are sensitizer, I put here that to show you that what pro-apoptotic binds with which. So you can BAD binds with BCL2, BCLXL, both HRK only BCLXL, NOXA and MS1, they can bind with MCL1. So here from these information, BH3 mimetic drug has been developed. So this is venetoclax, what I told already, that this is the only FDA approved drug for again BCL2. There are other compounds which are not drug, but they are S63845 with MCL1 and A-11 to BCLXL. So these information we are using further to not just only in drug discovery, but also in the clinical trial, that what drug means. Here you can see all are anti-apoptotic proteins, but they need different inhibitors to inhibit different anti-apoptotic protein. You cannot give means any inhibitor for all. So we must have to know that which patient or which cell is means what expression is high, which one. And so for that we are doing BH3 profiling. So actually BH3 profiling, this is the one of the very easy and lesser time taking approach to know and that which drug we have to give the patients. So, and also we will know this way that which anti-apoptotic protein is overexpressed. Excuse me. So here and this table just from these two tables from the last slide. So here it will be more clear that these BH3 only peptide, how they bind. So they can bind, BIM can bind with all anti-apoptotic protein, and bad can bind only BCL2. BCL is a HRK, the same what I showed last time in the last slide. So these BH3 mimetics means the inhibitor, small molecules. Here it is also here very clear, just like previous one. So we are using these information for BH3 profiling. So in BH3 profiling, so we are taking single cell suspension where the cell membrane is permeabilized, and we treat it with BH3 only peptides, any of these. So, and then after JC1 staining, we do the fluorescence kinetics, and from this we analyze the data, and then we can see that the particular cell it means which anti-apoptotic protein is overexpressed or more functional in that cell. So here DMSO there is no mitochondrial depolarization, and FCCP is a control. And I said BIM can bind with all. So you can see all BIM is also control you have to use to see whether how it is working. So and here you can see that MS1, it is the MCL1 dependent, because this is the high one. So this is the method to know that which drug should be administered to which cell line or which patient. So this BH3 profiling, I have done different cell lines of AML or leukemia. And that leukemia is acute myeloid leukemia. So in those you can see that, for example, this SKM1. So BAD is for BCL2, HRK is for BCLXL, and MS1 is here for MCL1. So in this BH3 profiling data can be proved by cell viability. So I took the SKM1 cell line and I treated with different known inhibitors, and you can see that S63845, this is the most potent, because S63845 is for MCL1. This is inhibitor, and here also you have seen that it is MCL1 dependent. So the same this way we confirm that BH3 profiling is one of the best technique to go further for preclinical trial. So in that we were using not leukemia, right? Because leukemia is general term. There are multiple type of leukemia. One of them is acute myeloid leukemia. So in that acute myeloid leukemia, there are two types of leukemia. One is secondary AML, one is de novo. So we are focusing mainly secondary AML. The reason is here. What happens that secondary AML has very worse response, and the relapse is very increased. So the difference, I want to tell you that what is the difference in secondary AML and de novo. De novo means first time it happens, this cancer happened first time. So that is de novo. But secondary AML means the patient was already suffering from some blood related disorder like MDS and other, or this patient has taken already chemotherapy for other disease or for other cancer. So that is so those patients are known as secondary AML. So the secondary AML, you can see here the survival graph. So this is de novo, the blue one, and rest all are secondary AML of different types. So you can see the survival rate is very low in case of secondary AML, because the cells are already they have either treated and all those. So and they are resistant for the chemotherapy. That's why the survival rate is very low. So we are focusing on secondary AML. So currently the standard treatment of secondary AML is chemotherapy, where cytarabine or Ara-C is taken as a DNA synthesis inhibitor. So the problem that if this chemotherapy is given to the patients, so there is the resistance. They develop resistance against this chemotherapy, and that resistance is because there is in high overexpression of BCL2 in secondary AML patients. So that BCL2 is anti-apoptotic protein which resists to kill the cell. So the cells are not killed even by the chemotherapy. So here you can see easily that in the basal cell. So this is BCL2 anti-apoptotic protein and the activator BIM. So it bound with this. So this is in the basal. So what happens when anti-cancer therapy is given? So there is very high expression of BCL2, and so all the activator which are responsible for apoptosis, they are bound with this BCL2. So what we do, we can add some BH3 mimetic, means which can target BCL2. Then these activator or BEAM or pro-apoptotic protein or peptides, they will be released and they will go and activate the inactive backs and back, and then apoptosis will happen. So this is our approach. So we are doing preclinical evaluation of already used chemotherapy with this venetoclax. Okay, so this is our approach, that how we are doing BH3 profiling, and after all the characterization, we also did to know that whether this drug cytarabine and venetoclax, whether they are showing synergy or not. So we can see there is synergy in secondary but not in de novo when we are doing cell viability. And you can see the early apoptosis. We did also apoptosis, and we saw there is huge synergy. So that's why we are using this one. And there is another way to know that dynamic BH3 profiling we also do. It is the same BH3 profiling, but the cells are treated already with the some drugs. So here, because the limitation of time, we have done, we are doing this thing, dynamic BH3 profiling, and which is confirmed by also Western blot, and by statistics there is significance. So we did also in the patient cell, and here you can see there is very clear synergy, and all those patient samples we put, this is a heat map of those patient samples of mitochondrial depolarization, and very clear that they are dependent, means what anti-apoptotic protein they are dependent. And this way our work is not just only drug discovery, but also if there is some known drug from the same kind of class, so how we they are used for clinical trial to the patients. So our finding says that yes, it is working, means we can use the venetoclax with the well-established chemotherapy. So thank you so much, and for your patience. So here is my acknowledgement. I acknowledge highly to my group leaders, Zaneta Nikolaska, and all our past members, and all our collaborators of the internal and external, and also to our fundings. Thank you so much. If there is some question. So you are open for the question. Thank you so much.
Yes. Hello.
Okay.
It's a really wonderful talk. Actually, we have one doubt. How BLI method is better than other biophysical interactions?
Okay, so it is not better. It is the, because every method, every approach has their own limitation and their own benefit. So you can say there are always pros and cons of each method. So BLI, actually, this is the technique which I like, which I don't like much. But because if I have to do some BLI, I will go to surface plasmon resonance. But anyway, so BLI, you can do high throughput. So high throughput screening, if you want to do, you can use BLI.
Okay, thank
So
you, sir.
when you can use 96 plate 84, but in ITC another you cannot do it. So it is a little bit easier, and you can say this way that it is high throughput. For drug discovery, you have to be more, means any method what is high throughput that will be feasible for any drug discovery.
Hello, sir. Thank you so much for the wonderful talk. I just have a quick question, like a drug resistance regarding the cancer treatments. How we have to, focus on the future, because there are so much of drugs that are getting resistance to the cancer cells, and even actually I'm working on AMR, antimicrobial resistance. So same thing everywhere, drug resistance. So how far, how we are to focus on the, or how to tackle these drug resistance issues?
Yeah, so this is really challenging, because so yes, so this is the study that what we are doing, because in our case we know that what is the reason of the resistance. So we have to target that one here. The for the chemotherapy, the the reason for drug resistance is BCL2 overexpression. So we are inhibiting that protein to overcome. So this is the way we have to find out what is the key means protein or gene which is responsible for, and then you have to target. So you can see here chemotherapy, and then the resistance because of chemotherapy, we are targeting also that. That's why we are using dual aim.
Okay, sir. Okay, sir. Thank you. Thank you so much.
This is the only way we can do.
Thank you so much, sir, for your enlightening talk. We will now proceed with our next speaker, Dr.
Thank
Shankari
you, sir.
Nagarjun. Dr. Shankari Nagarjun is a dynamic professional with a rich, diverse background in molecular biology, cellular function, and cancer research. With a PhD in molecular biology of cells from the prestigious Göttingen Center of Molecular Biosciences in Germany, she has honed her expertise in unraveling the intricacies of cellular mechanisms at the molecular level. Throughout her career, Dr. Shankari Nagarjun has held pivotal roles at renowned institutions worldwide, including the University of Cambridge, where she served as a senior research associate at the Cancer Research UK Cambridge Institute. Her contributions to the Division of Molecular and Cellular Function at the School of Biological Science at the University of Manchester have been instrumental in advancing our understanding of chromatin biology. Dr. Shankari Nagarjun's research journey extends beyond borders, encompassing diverse experience as a postdoctoral fellow at Department of General, Visceral and Pediatric Surgery at the University Medical Center of Göttingen, Germany, and as a junior research fellow at both the Rajiv Gandhi Center for Biotechnology in Trivandrum and the Stem Cell Biology and Regenerative Medicine Division at the National Center for Biological Sciences at Bangalore. Her research findings have been disseminated in numerous peer-reviewed journals reflecting her expertise and innovation in areas such as chromatin biology, cellular function, and cancer progression. In addition to her scholarly achievements, she has been honored with several prestigious awards and accolades, acknowledging her outstanding contribution to scientific inquiry and academic excellence. With a wealth of expertise spanning in lecturing, research, and project management, she brings a unique perspective to her work, blending scientific rigor with a passion for discovery. Dr. Nagarjun is poised at the forefront of cutting edge research in cancer biology, driven by a relentless pursuit for excellence and a vision of a world free from the burden of cancer. Please join me in welcoming Dr. Shankari Nagarjun to the podium with a warm round. Thank you.
Thanks for the great introduction. Can I just make sure you can hear me okay and you can see my slides?
Yes, ma'am.
Yeah, thank you. That's a really great introduction. I'm quite impressed. Thank you so much for that. So I will start with how chromatin biology work in case of breast cancer. So it is quite complicated mechanism. So I'm hoping that I can explain it in a simpler way. So I work in enhanced biology and epigenetic factors. So epigenetics really means anything other than DNA if it can influence any cellular processes like phenotype. So I majorly work on epigenetics, and especially I work on this particular DNA regions called as enhancers. Enhancers are regions which are really far away from gene promoters. So if you know, gene promoter is really, really important for transcription. And this particular enhancer region is quite far away in case of, if you think about the primary structure of DNA, it is really far away. But there are proteins, complexes, and they work together to bring them together. For example, you can see, let me have a mouse pointer option. Yeah, so you can see proteins like cohesin complex, mediator complex, other transcription factors. You can see mediator complex here. There are a lot of different protein complexes which will bring these DNA regions closer to gene promoter, and these factors are very important to drive transcription. So also factors like SWI/SNF complex. This is a chromatin remodeling complex. It remodels the chromatin, and that is very important to bring the chromatin together, like the enhancer and gene promoter regions together. And we also have general transcription factors, which will start with the transcription. But don't forget about the super elongation complex. This is important to have the transcript completely formed, because if we start the transcription, it can get paused around 40 nucleotides. So it is very important to have the super elongation complex to have the whole transcript to be transcribed. So you can see that it's multiple complex based mechanism, and transcription is not very easy, and enhances work in case of any stimuli. Like if you need to treat a cell with hormone or anything, you need actually enhancers to enhance the transcription, and that's what it is. So we work on nuclear receptors in case of breast cancer biology, especially a protein called as ER, or estrogen receptor. And this estrogen receptor is the major nuclear receptor in case of breast cancer. Around 70% breast cancers are positive for ER. And this ER works along with this factor called as FOXA1. FOXA1 is a pioneer factor. That means this factor is needed for ER to actually bind. So FOXA1 can bind to even chromatin when it is condensed, and it will try to open up the chromatin with the help of the SWI/SNF complex, because SWI/SNF complex is the chromatin remodeler, and together they function to open up the chromatin, and ER can bind there. So this is a complex I majorly work on. And from my PhD, I have done a lot of work in this transcription mechanisms, especially in super elongation complex, a protein called as BRD4. It is known to have role in elongation and also viral transcription. But with my study, I identified that this is important for estrogen receptor dependent enhancer activation. So basically, even though it binds to promoters to have the transcription completely done, it is also important for enhancers. So it is also bound to enhancer regions and regulate estrogen receptor dependent transcription. And that was my PhD work. And I also published work with FOXA1, where we know that enhancer recruitment in like proteins like MLL3, which are important for activating the chromatin. So it is needed for enhancer activation, and FOXA1 drives them. But my recent work is about ARID1A. It belongs to the SWI/SNF complex, chromatin remodeling complex. And there I have integrated all these factors, basically ER, FOXA1, and BRD4. And we identified that ARID1A is important for BRD4 and HTAC1, like deacetylases, to bind, and it activates. So it basically controls treatment response. So they are important for tamoxifen response, for example. Tamoxifen is a drug we use to target ER, and ARID1A is important to control that response. And with my new work started in University of Manchester as a lecturer and group leader, I'm funded by Cancer Research UK, where I'm trying to also establish how these other proteins like cohesin complex, mediator complex, how these are involved in drug resistance, because we have worked with ARID1A, which is involved in drug resistance, like the loss of that protein leads to drug resistance. And now we are also working on these cohesin and mediator complex, whether that is also involved in drug resistance. During my PhD, I worked with BRD4, but I also identified that this particular protein, it can regulate non-coding RNAs. And these non-coding RNAs, not link RNAs, but they are transcribed out of these regions of enhancers, and they are called as enhancer RNAs. And enhancer RNAs are important for enhancer function as well. So I'm going to talk in my first part of my seminar, I'm going to talk about this enhancer RNA a lot. So enhancer RNA, they're important for enhancer activity. So as soon as ER alpha binds to the enhancer region, these transcripts are coming out, and they don't translate any proteins. They're just non-coding, but they are important to have the transcriptional mechanism like functional. So they're important for the chromatin condensation to happen, like the enhancer and promoter region to come together, and like also stabilizes binding of cohesin protein and mediator protein. So without enhancer RNAs, the activation by enhancers is not happening. The thing is, enhancer RNA depends upon the enhancer size, like how long they are transcribed. So if the enhancer is 1000 base pairs, it can be 1000 base per transcript. If it is like 10 kb enhancer, of course, it can be 10 kb enhancer. The median size is 335 base pairs, and they are early indicator of estrogen receptor activity. As soon as this enhancer RNA transcript is made, enhancer activity is happening, and they are needed for mRNA production. Like whichever gene is close to them, they start regulating their expression. This is identified in a means of knocking them down or doing antisense oligonucleotides, or if you put a transcription termination site in this enhancer region, we could see that mRNA transcription is also affected. So this means that the enhancer RNA are important for mRNA transcription. The issue with studying these enhancer RNAs, you can see here, enhancer RNAs half-life is really low. They are the most unstable RNAs in the world. You can see mRNA has around 100 minutes of half-life, but generally enhancer RNAs have only 2 to 3 minutes half-life. So what, like you would be knowing about how mRNA can be isolated. We always need to be really careful in doing RNA isolation. But in case of eRNA, it's really not possible to detect them. It's quite difficult. So you need to really have your bench clean. And even in that case, in 2 to 3 minutes, they are going to be degraded. So it is really hard to develop techniques to detect them. We have done multiple methods, especially GrowSeq. It's a global run-on assay. So you basically isolate nuclei, and then you do in vitro transcription, and then you can isolate them by either biotinylated residues or fluorescent tagging, all these things. But they don't work all the time. The reproducibility of the experiment is only 50%. So it's very hard for people to establish these methods. RT-PCR is easy, like you can do qPCR, like after isolating the RNA, you have to use random primers because these enhancer RNAs are not polyadenylated. Only 10% of enhancer RNAs are polyadenylated. So the interesting ones are other 90%. So you need to use random primers when you do reverse transcription. And the problem is you can do only one by one, right? One each eRNA per PCR. So you can't do like a sequencing to detect all of them. And the same thing runs with RNA fish. But the problem is it's a laborious technique and low throughput. It might be challenging for enhancer RNAs because of their labile nature. They are unstable. And when we prepare tissue samples, for example, like if you want to detect enhancer RNAs in tissues, it's really hard. But we made it work. So in collaboration with Cambridge Institute, funded by CIUK, Julia Jones, who's a really great expert in RNA fish, we have published book chapters together with her. So she developed this particular method using RNAscope from Biotechne Advanced Cell Diagnostics Company, and we could detect enhancer RNAs, which was really amazing, helpful. So I'm going to show you one particular enhancer RNA here. So for example, this particular gene GREB1 is a very nicely studied estrogen receptor induced gene, and it is important for breast cancer proliferation. And you can see the transcription happening here, but also there's the ER alpha binding site, which is quite far away from the promoter. And it has all the active enhancer marks, like they are producing non-coding transcripts. You can see as the around the intensity of mRNA, it is also producing the eRNAs. BRD4 is bound there, enhancer mark, and again a K27 acetylation is an enhancer mark. These are all heavily bound on this region. So what we did, we tracked this transcript, which is getting produced, and we designed a probe against it for RNA-FISH or RNA-ish. And this MYC is a famous estrogen receptor targeted gene. It's also involved in leukemia, for example. MYC is like everywhere. It's important for proliferation. This has multiple enhancers. As you can see, ER alpha is bound in multiple regions. So we designed multiple probes, like around 3 probes to detect it. So you can see like so much of transcription happening, like non-coding RNAs coming out of this. And we designed different probes for that. So this is an example picture for detecting the enhanced RNA from GREB1. So you can see that like nice dots, which are RNA. So a bigger dot means there are multiple RNAs. They are coming together from the same allele, same spot in the transcription. There are also smaller spots you can see. So we can see the enhanced RNAs really nicely with our RNA-FISH method. And this is an antisense probe, which means it shouldn't work. It's not detecting any RNA. They are like a negative control for this. And this is the example pictures for MYC enhancer region. So you can see like nice dots coming out of them as well. Like it's amazingly detecting the RNAs. To make sure that this is real, this is really enhanced RNA, we designed locked nucleic acids. So basically like antisense oligonucleotides to stop the production of the enhanced RNA. And we quantified the enhanced RNA with RNA-ish, and you can see the locked nucleic acids are great effect. In the control locked nucleic acid, we do see nice expression of enhanced RNA, but we don't see that with the locked nucleic acid. We tested this with 2 different cell lines of ER-positive cell lines, MCF7 and T47D, and we could see the same effect in both the cell lines. And then we started asking questions about, okay, we know that we can detect this now, but then if we target estrogen receptor, would it affect the eRNA? Like, is that sensitive enough to actually replicate what's happening in transcription in case of targeting estrogen receptor? So estrogen receptors can be targeted in multiple methods. When the drugs are available in the clinic for long time, aromatase inhibitors, for example, they stop the production of estrogen. But anti-estrogens like tamoxifen or Faslodex or fulvestrant, these are estrogen receptor modulators or degraders. They stop the binding of ER to the chromatin. They don't stop the binding of ER to the chromatin, but they stop the binding of estrogen to the receptors. That means that the receptor is not active, but the receptor can still bind. But tamoxifen will be replacing, like competitively inhibiting estrogen binding to the receptors, so there won't be any cancer cell growth or transcription happening. So here is a analysis where we did estrogen treatment, where you can see different time points. Even in 2 hours of estrogen treatment, we saw a huge amount of enhanced RNAs getting transcribed, but in 24 hours, it's even more, like it's getting stabilized in 6 hours. But tamoxifen and fulvestrant, both of these drugs, actually stop the production of enhanced RNAs like quite significantly. Either you do them with hormone deprived condition and treat the cells, or you just do it with full media, like complete media. Don't starve the cells, just treat them with the drug. It works amazingly well. That means the eRNA detection is quite sensitive. This is an example with EMIC enhanced RNAs. We have designed 3 different probes, but you can see that the first 2 are not really responding much other than 24 hours. It's getting all destabilized. But we do see nice regulation with the 3rd probe, where you can see in 2 hours, they are working nicely and then they are going down. So then we started testing them with tamoxifen resistant model. So we have a model called PDX model, a patient-derived xenograft, in collaboration with Alex Bruno and Carlos Caldas. So he's a big shot in breast cancer research. So he have developed a lot of PDX models so that we can test our drugs and everything. So we got explants from these models, especially STG195. This model is estrogen receptor mutant, so they don't respond to tamoxifen much, and we all know that. And we could see even in this tamoxifen resistant model, like we take the tumor, We cut them into small pieces, and then we put it in a dental sponge. So for 2 days, we can track the response to a drug. After 2 days, they start dying because they are in a sponge. They are not in a live condition anymore. But in the 2 days, we can see any response, and we can do any kind of treatment. It's a cool method. So what we did treatments on this dental sponge, and we can see that in the tamoxifen resistant model, all these drugs are enhancing the production of eRNA. So this could mean that this eRNA is getting upregulated in the tamoxifen resistance or fulvestrant resistant model. Could also mean that this is probably having some role in drug resistance, like promoting it. It can't be seen with the GREB one. It's just the overall regulation level. Even with enhanced RNA, we can see a nice dynamic regulation, and this is probably activating everything. In a TAM-sensitive model, another PDX, we don't see that, but we see it specifically with tamoxifen-resistant model. Enough with the therapy part. So now I'm going to the biological mechanism behind this. And we want to really understand the upstream regulators of enhanced RNA. And we developed our RNA-ish method to RNA-fish, where we can also multiplex multiple RNAs. For example, in this assay, we have multiplexed the enhanced RNA, the corresponding mRNA, and also a proliferation mark, which is Ki67. So if a cell is present with Ki67, that means the cell is proliferating. So this will tell us not only about the upstream regulators of mRNA, but also the eRNA, and also what happens to proliferation. So when we did this, like we can see the green color with enhanced RNA and the red color, the mRNA, they are co-localizing with each other. So they are coming from the same active transcription hub. And you can see mRNA is moving to the cytoplasm, which should happen normally. And also we can see the Ki67 mRNA moving to the cytoplasm. They are all mRNAs. They have to move to cytoplasm. But enhanced RNA is usually nuclei. But you can see small dots in the cytoplasm as well, which we don't know the function, but it's usually nuclear. What we developed was, based on this method, we established a CRISPR screening where we knocked out 234 proteins, which involves 150 epigenetic regulators and 70 core interactors and also nuclear receptors. We made guide RNA viruses for them. So in case of like arrayed libraries, so each well will have a one gene knockout. So we can also do the knockout on the cells for each gene, and then we see what is happening with the eRNA, mRNA, and also the proliferation. So it's a massive experiment, also very expensive experiment, because we made CRISPR screening, we made CRISPR viruses, and we also did RNA-fish for 9 plates. And we have to do high content screening using a microscope called as Operetta. So we can do live screening as well with that. We did only fixed cell screening, but we got like confocal like images of like 1,000 cells per well. So it is such a huge experiment. So we did only 2 independent replicates for that. So basically, we wanted to address what are the upstream regulators of eRNA synthesis and what it does to mRNA transcription, as well as whether it affects proliferation. We did have batch effect, but you can see it's only a few of the genes which have the batch effect. So basically, this is rep1 and this is rep2. So the yellow one is rep2 and pink one is rep1. So you can see this batch effect only on few of the genes, but most of them have similar effect. So you can see different effects as well. So starting with it, all the co-repressor complexes came in the red part. That means increasing gene transcription after the knockout. And all the ER complex, like even ER alpha, ESR1 is ER alpha, FOXA1, all these important proteins came in the bottom part, which is blue. Blue means it's down-regulated expression after the knockout. So they are important for the gene expression. And this all comes very nicely. So hashtags or histone deacetylases, they usually inhibit transcription. So that's why when we remove the protein, we saw like upregulation in gene transcription. Interestingly, we saw this Weisneff complex, chromatin remodeling complex, which I've worked before on, came up where most of the subunits of the Weissenhoff complex are upregulating gene transcription after the knockout. So they are basically co-repressors. We also saw a protein ARID1A, which we have done a lot of work on, and that had kind of batch effect. One replicate showing nicely upregulated gene transcription after knockout, and the other replicate didn't. So we tried to look at this, and we could clearly see that there's upregulation in gene transcription, either in a xenograft experiment, RNA fish experiment, or a qPCR with reverse transcription. You can see the red one is the ARID1A knockout clone. So we made like various clones for it. And the different, even not only GREB1 related enhanced RNA, but different enhanced RNAs are upregulated by ARID1A knockout. The other thing we saw as a top most hit in our screen is a surprising protein, which I have never worked on. It's called as BRD1 or BRPF2. This belongs to a complex called as MOS-MOF or HBO1 complex. It's in the histone acetylation complex. So they basically I hope I have a model here. Yeah. So basically, this complex either acylates H3K14 or H4 acetylation. Depending upon which factor is bound, the acetylation can be reverted. So we got a hit on BRPF2. This is usually H4 acetylation, but it can also acetylate H3 if it is associated with MOS-MOF complex. So it depends upon which histone acetyl transferase is there. Interestingly, this is promoter-based histone acetyl transferase complexes because they have this protein called ING4R5, and they are recognize only H3K4 trimethylation, which is a promoter mark. So they can't really go to enhancer. But in our screen, we saw that it is regulating enhancer RNA. So it was quite surprising to see this effect. And we saw that it's enhancing the gene expression, but MOS-MOF is down-regulating gene expression mildly. HBO1, which is another histone acetyl transferase, it's having a mixed effect. So we don't know what's really going on. So we wanted to explore this. So first thing we did was first validate our screening. So we did it with eRNA qPCR, RT-qPCR, and we could see that the knockdown of BRD1 actually induced enhanced RNA transcription, but it's not happening with all the genes like ARID1A, but it's happening only with few of the genes. Like MYC has a bit of an effect, but eGREB1 has only slight effect. So we wanted to know what's really happening. So then I started my group after this work, and my postdoc jumped in to help me with this project where she did a lot of ChIP-seq analysis. And ChIP-seq means we cross-link the chromatin, and then we look for protein occupancy. And if there is a darker color of anything, means that particular protein is bound there. So she did analysis on this BRD1 protein, like where it is binding to promoters, and she could clearly see that it's mostly binding to promoters. You can see like around 61% of them, or even included like more than 90% they are binding to gene and gene promoter, gene, little bit into gene. It's not binding to enhancers. And we could see like HDAC1 is also nicely bound there, and this is heavily marked with promoter marks. And this is the acetylation happens with BRD1, so that's also a little bit there. Polymerase is binding there, so RNA polymerase, which is important for transcription, is also binding there. So we could see them located to promoters, but they are not binding to multiple regions. They are binding only to few sites. So those genes are important for estrogen receptor transcription. Basically, they are ER complex co-activators. So that explains it, because if it is binding to estrogen receptor complex proteins, the genes of them, maybe they are regulating the transcription of these genes, and that's why it is regulating enhancer RNA, even though they don't bind to enhancers. So it's quite indirect way of doing it. So for example, it is binding to GREB1, that means it can regulate ER complex activity, that means it can regulate an enhancer RNA. So it is quite really indirect mechanism. So here I'm showing you with GREB1, you can see BRD1 nicely bound in the promoter region. So basically starting from this particular gene promoter, not even the other ones. They are not binding to enhancers. Like you can see enhancers here where ER binds, they are not binding there. They are binding close to the K4 trimethylation regions and polymerase, like wherever we have the acetylation. And we could see that the knockdown of BRD1 induced the acetylation. So we thought they are important for acetylation. The acetylation will reduce, but they have other effect. They are inducing H3 acetylation, but that makes sense because they're inducing acetylation. That means inducing transcription. So that makes sense. But doesn't make sense is we thought they're important for the transcription, but they are for the acetylation, they are actually really controlling them. So maybe we believe that maybe it is activating the knockdown of BRD1, activating this particular acetylation. Maybe it is having a reverse effect on H4 acetylation, which is something we are still exploring. So the summary was, so we have established a method to detect really unstable RNAs in cancer cells. We can also detect them in patient samples where we can, if they are fixed FFPEs, we can detect them really nicely. And we have used CRISPR screening to detect the upstream regulators of them, and also they regulate proliferation. So basically ARID1A, BRD1, all these are controlling enhanced RNAs, but also ER complex proteins, they are promoting RNAs. And I have not shown this data, sorry about that, but the regulation in eRNA, it also controls proliferation. So they're very nicely correlated. Whichever proteins are regulating eRNA, they are also regulating proliferation. So we believe that this BRD1 is probably controlling eRNA expression, either by regulating the expression of the ER complex coactivators. They are not binding to enhancers, and regulating histone H3 acetylation. So if I have time, I can quickly move to this another story. Like if you can confirm if I have time. I mean, it might not take, I think it will take 10 minutes if that's okay. This is probably more related like cancer wise.
Hello.
Yeah.
Ma'am, please stick with the time.
Yeah. Or shall I talk about the other story, or I can stop otherwise?
You can brief it, ma'am.
Yeah, I can quickly go through it. Thank you. So this story is about triple negative breast cancers where this is not having the receptors like estrogen receptor, HER2 receptor. None of them are there in these cancers, but they are really aggressive cancers and they are highly metastatic. So we really want to know why this cancer actually is like the survival rate is really poor. Like most of the patients die, and the patients are diagnosed like in 30 and 40s and they die at that age. So it's really unmet need cancer to be studied. Recently, we could see that SWI/SNF complex is heavily inactivated in these cancers. You can see like they're also metastasis specific. Like mutations in ARID1A, ARID2 within this complex, they are representing only in the metastatic patients, not in primary patients. So this is really interesting for us. So this SWI/SNF complex overall is like it is different complexes. You can see like BAF, which has ARID1A, PBAF, which has ARID2, and PBRM1. These are important proteins, and this one is not much important. But these can replace each other as well. What it does for SWI/SNF, as I said, is chromatin remodeling proteins. So it can open up the chromatin for a transcription factor to bind. And so that histone acetyl transferases can come and bind, and then they will activate the chromatin and the transcription can happen. What we saw was with the triple negative breast cancer, when we lose PBAF, like by mutations, the survival rate is really poor. And this is very interesting because when we knock down ARID1A in triple negative breast cancer cell lines, like 2 different cell lines, nothing happened. But ARID2, which is the PBAF complex protein, there's huge amount of migration happening in cells. So if we remove the PBAF, the cells are becoming more aggressive. We wanted to understand this. We tested it with different cell lines and also different subunits of the complex. We could see like the migration is really high when we remove PBAF subunits, and it is more represented in cell lines which are more epithelial, like MCF10A or BT549. You can see significant effect. SUM159 is a cell line which is a little bit mesenchymal, so we saw effect, but it's still milder. But removing them increases the migrated capacity. So then we thought of doing like what happens to the chromatin. So when we remove ARID2, we could see both repression and activation, but repression is slightly more, and we could see that in the active enhanced mark as well, like repression is slightly more. It's not only just activating chromatin, it's also repressing chromatin. And what we did was we took these repressed regions and then we mapped with the ATAC-seq and we mapped them for activation. We saw the same effect. We just classified them with repressed region, but we could see the same repression. Like you can see, like the knockdown is repressing. So basically, it is having more activation. And these regions, which are bound with both ATAC gain, basically the repressed regions, the gain regions, and those are 312 sites. And we did pathway analysis, and we could see all the breast cancer related pathway and also highly aggressive pathways like EMT and EGFR signaling. All these are heavily up. So what we did was we did wanted to identify what factors which are binding here to really activate this aggressiveness along with ARID2. So we did motif analysis and we saw particularly interesting proteins like VDR and GR. These are nuclear receptors because transcription factors can't be targeted, right? So we want to know about some factors which can be targeted, and nuclear receptors, all of them have targeting agents. We saw the same here, like with different motif analysis, we saw multiple nuclear receptors, including GR, and RXRB is VDR complex protein, like they become heterodimers. This GR and VDR story was interesting. So what we did was we inhibited GR, or we activated GR, or we inhibited VDR. We could see that VDR inhibition actually stops that aggressiveness, like the induction in migration is stopped. Activating GR, it's you can see like it's activating it more, like activating GR is really bad, but inhibiting has a mild effect. And this is just preliminary data. And we could see that the repressed regions also have GR binding, which was clearly like visible. You can see like they have even induced binding in these regions. So our hypothesis is that this PBAF complex is probably repressing nuclear receptors, and when the cancers have loss of PBAF, these nuclear receptors are highly active and they can activate migration and aggressiveness in triple negative breast cancer patients. Then this is probably easily treatable because there are inhibitors available for nuclear receptors, like even GR, and hopefully we can solve this more. Like this is just a work in progress, which I wanted to share. We could actually identify the nuclear receptors. Now, right now, we have some work on glucocorticoid receptor, which is VGR, and vitamin D receptor, and we hope we can expand the study because we believe that ARID2 can repress GR and VDR, and hopefully we can use the inhibitors to stop this migration or metastasis. With that, I will conclude. My great acknowledgement goes to the group in CIK, Cambridge Institute, the University of Cambridge, and all my collaborators and my group where Sumitra, my postdoc, she did a lot of work with BRD1, and Simone, our research assistant, and the triple negative breast cancer story is from one of our PhD student. And I would like to thank all my funders, Cancer Research UK and Breast Cancer Now, and also the University of Manchester. With that, I will conclude my talk.
Thank you so much, ma'am, for sharing your expertise with us. Now we would like to have some questions. The first one being here, could you please elaborate on the functional diversity of eRNAs in different cellular contexts?
That's a very interesting question. So what we see is like any kind of enhancers, doesn't matter it's during development or cancer or metastasis, we do have enhanced RNAs. And there are a lot of enhanced RNAs, they are getting implicated in treatment response, treatment resistance. So I believe that enhanced RNAs generally are context specific. They are driven by transcription factors, and whatever cell processes are regulated by transcription factors, they will all have enhanced RNA. So they are quite ubiquitous, I would say, but they are specific to a transcription factor. So you won't find, for example, you won't find the same enhanced RNA present in another cell, for example, like breast cancer specific enhanced RNAs, they won't be expressed in a different cancer. So that is known because the enhancers are different, even though they are conserved, but the enhancers are different. The driven enhancers are different. In that case, they are context specific.
Thank you so much, ma'am. We have question and answer segment open for both Dr. Shankari Nagarjun and Dr. Ejaz Ahmed. We invite students to actively participate by posing their questions to our esteemed speakers. Ma'am, this one is again for you. How do genetic and epigenetic alterations influence the enhancer RNA activity?
That's a very good question, and I would say it's gonna be a project in my lab. So what we believe is, in case of cancer, the genetic aberrations in proteins like epigenetic genes, for example, SWI/SNF complex proteins, they are heavily mutated in pancreatic cancer or esophageal cancer and multiple cancers. They are after P53, ARID1A gene is the second top most mutated gene, like in case of tumor suppressors. So that is a genetic modification, right? A genetic perturbation happening, and that is happening on epigenetic complex protein gene, and that is the epigenetic regulation, and that's what we believe, that it's regulating all the cancer. So this genetics and epigenetics is quite intertwined, by the way, like because it's happening on the epigenetic complex proteins, it is affecting the epigenetics. So, and people also try to work with this epigenetic complex regulating genetic alterations, because ARID1A, for example, it's heavily involved in DNA repair and genome instability. So a genetic modification can cause epigenetic changes, and then this epigenetic changes can cause genetic changes. So it's like quite cyclically happening. So this is something we really want to establish in a different project, where ARID1A, like the mutations in ARID1A, what it does to epigenetic complexes and what it does to genome instability. And that would be a really interesting study.
Thank you so much, ma'am. This one is for Ejaz, sir. So, what are the strategies that are being explored to enhance the efficacy of the BH3 mimetics for overcoming resistance mechanisms and in cancer treatments?
Yeah, so as already I had shown all those slides, so those are related with this question, that because of the chemotherapy, there is a resistance. So resistance is because of the BCL2 family proteins. So we diet inhibit that one. So that's why there is a need of means to get more drugs against those BCL2 family proteins. So only BCL2 family has FDA approved, but still there is scope for MCL1, BFL1, BCLXL. So we had to find out that which protein is responsible for that resistance, and finding out the inhibitor for that one. This is the only solution, and only means the way to get the resistance. Yeah.
Thank you, sir. This is the last question. This one is for ma'am. Ma'am, how can epigenetics be used in different cancer diagnostics?
Oh, that's a great question as well. So this is something I'm working on. Maybe it will be a long-term plan, where I propose that enhanced RNAs can be used for diagnostics, even early cancer stages, because eRNA is an early event of transcription. So we believe that if we detect the proper or right enhanced RNAs, which are heavily expressed, for example, in a particular cancer type or particular cancer stage, so we could detect for example, metastasis, because we know like there are metastasis specific enhanced RNA, so we can really develop it for like diagnostics purposes. Especially in this case, RNA-FISH will help because RNA-FISH is something we can do on patient samples like biopsies. And you can probably also detect these RNAs in circulating tumor cells. That means you can probably detect them in blood, but this is really, really long-term plan. So I believe that in this kind of scenario, we can use epigenetics as a diagnostic approach. But people recently started doing a lot of DNA methylation in circulating tumor cells in case of metastasis. And this can be developed, like liquid biopsy based research in epigenetics can be definitely developed sooner as for diagnostic approaches.
Thank you so much, ma'am. As we come to the end of our enlightening seminar on emerging trends in cancer biology, we would like to extend our heartfelt gratitude to each and every one of you for participation and engagement. Today, we had the privilege of delving into the latest advancements, breakthroughs, and discoveries in cancer biology. Thanks to the insightful talks and discussions and invaluable insights by our esteemed speakers, Dr. Ejaz Ahmed and Dr. Sankari Nagarjan. On behalf of the Center for Discovery and Development, Series 3, Amity University of Biotechnology, I thank you all. Lastly, can I please ask everyone to switch on their cameras so that we can take a picture?
Taken?
Thank you so much, everyone.